Good afternoon and welcome to the Human Brain Project Platform Release event. I'm Philippe Gillet, I'm the Provost of École Polytechnique Fédérale de Lausanne, with the coordinator of the Human Brain Project. This event is being streamed live via Adobe Connect and YouTube to viewers all around the world. Part of the program is taking place here in Geneva, in Switzerland, but we will also be joined by HBP platform teams connecting from sites from Germany, the UK, and some places in Switzerland. This platform release event is a very important milestone for the Human Brain Project. Today, we are pleased to announce the opening of the prototype versions of our 6 platforms to the scientific community worldwide. These platforms are Neuroinformatics, brain simulation, high-performance analytics and computing, medical informatics, Neuromorphic Computing, and Neurorobotics. This release comes after a 2.5 years period of time after the launch of the project. The HBP is one of the 2 FED flagship initiatives funded by the European Union's Future Emerging and Technology Programme, which is part of the ICT arm of the EC, and it is funded for a period of 10 years, and we are extremely grateful to the EU support, but also from the support from national governments throughout Europe. The HBP platforms have been developed through a highly collaborative effort involving more than 750 scientists and developers from 114 institutions in 24 countries, primarily in Europe, but also in the US, China, and Japan. These platforms embody the key objectives of the HBP, namely gather and disseminate data describing the brain, simulating and building models of the brain, developing brain-inspired computing and robotics, and creating a global scientific community around the developing research infrastructure. They are, of course, not finished products. Today, they consist of prototype hardware, softwares, databases, brain atlases, programming interfaces, and initial data sets. The level of maturity, as well as access to tools and features, differs from platform to platform. Some of the tools we have seen this morning during our video conference with the potential users, have been built on earlier projects and are thus in a more mature status than others. This is, for instance, the case for the Neuromorphic Computing platform. Other tools, such as many in the medical informatics and Neuroinformatics platforms, have been developed at the very beginning of the HBP. This means 2.5 years ago. So they are in their infancy, but they are still operative. In some cases, we need to have access to very high-performance computing systems. This is done because we need to have high-performance computing to deal with the models and the simulation of the brain. Starting today, we have also warmly welcomed the wider scientific community to begin to use these platforms and to help us improving them. While the first phase of the project, the last 2.5 years, focused mainly on building the platforms, the next 7.5 years will focus more on user engagement and building a user-centric research infrastructure for brain research. We would like to work with the scientific community, the users of the platforms, but also potential other users, because this platform will be open widely. But we need the inputs to refine of these users to refine and adapt the platforms so the HBP can become a truly relevant infrastructure for brain research, helping to facilitate the development of a common understanding of the human brain. So this afternoon, we will give you a few insights on the different platforms. We have a program and we have an agenda for this program. So I give you a short. This was my short introduction. After that, I will give the floor to Catherine Amunds, who will present the significance of the platforms for brain research. Jeff Miller will give you a few insights on the way you can connect to these different platforms. And finally, we will have a brief presentation of all these platforms by a series of presentations, which will last only a few minutes each of them. And finally, we will open the floor to the questions from the journalists And in the end, we will give a few final conclusions to this afternoon event. So I think I will give the floor now to you, Catherine. Where are you, Catherine? Ah, you're here. Okay, so good luck.
Thank you, Philip, and welcome everybody. Good afternoon. The Human Brain Project aims to contribute to a better understanding of the human brain, and this is not only a question for basic neuroscience, but also for clinical research and a prerequisite to improve diagnostics and therapies for patients suffering from brain diseases. The human brain, however, is also one of the most complex structures, with its 86 billion nerve cells, the same number of glial cells, 10,000 synapses, and of course every single cell is also a complex organism and acting, for instance, through transmitters and their receptors in a very dynamic way. So to tame such complexity, we have structured the neuroscience research in the HBP into 4 sub-projects: mouse brain organization, human brain organization, systems and cognitive neuroscience, as well as theory. These 4 sub-projects contribute and benefit from the 6 platform projects which have been introduced in the morning, and you will hear about these platform projects more in a minute. Ethics represents an independent sub-project and interacts with all the other sub-projects. And that it is an independent project shows in importance, and this is quite unique for a project having such a big ethics sub-project. We have also introduced in the operational phase, which starts this week, so-called co-design projects, where neuroscientists on the one hand and developers closely collaborate to build the platforms and to make them usable and nice for the applicants, for the neuroscientists, for instance. So the way how we conceptualize cognition and processing of emotions or behavior is very much related to the way how the brain is segregated. Functional neuroimaging can be used to identify networks of areas contributing to a certain brain function, and they also help to identify dysfunctional networks, for instance. Here are shown some examples from Bertrand Thirian's lab, which show activations evolving during cognitive functions in subjects in the scanner. These images can be integrated into the human brain atlas and can be analyzed with respect to the localization in the human brain atlas. Another level which is highly relevant for brain function relates to the activity of neurons, and Hype Mansfelder from the Human Brain Organization team and his group compare neurons between mouse and human brains, and this is a necessary prerequisite to better understand the similarities but also the differences between both species, and to identify parameters, for instance, which can be used from the mouse brain studies for analyzing the human brain. Yedan Segev and colleagues from the theory sub-project and the neurosimulation platform, they have developed computational methods for these pyramidal cells. And most interestingly, these simulations provide new data that help us to do then again empirical experiments and to approach the differences between the morphology and the physiology of human and of mouse pyramidal neurons. Another level which is highly relevant is, of course, a microscopical level. And until now, it was not possible to map data coming from single neurons, for instance, like those which I showed you, to the human brain atlas, because no atlas was available until now due to limitations of the spatial resolution. In addition, most of the existing atlases are referring only to one single modality. Here we have developed the Human Brain Atlas, which is a true multi-level and multi-scale atlas, and which allows to map to the brain, considering the topography of the brain, single-cell data, molecular data, functional imaging data, patient data, and other type of data. And this makes the Atlas multiscale and multi-level. Of course, we can only start to populate the brain because brain mapping is very time-consuming. And to map a whole human brain, this is out of reach of a single research lab. Instead, we should engage the whole community, and the Human Brain Atlas provides a tool that people, researchers, can exchange information and contribute to a better understanding of the human brain. But this, at the end, also brings me to the problem that when we address the human brain on all these different levels, we end up with lots of data. And to process such data, like the connectivity data here, from the micrometer scale to the centimeter scale, is only possible when we use high-performance computing, and the simulation platform here was indeed very helpful. And what you see here are single nerve fibers which can be followed over large distances. So all these examples demonstrate, in my view, that already now the neuroscientists in the HBP have already a lot of benefit from these platforms. And I hope that you can join us and explore the new possibilities, try out what the collaboratory brings and which new possibilities it opens to analyze the human brain. And of course, this is not an easy process, and sometimes it is different to match the different cultures, and it's not easy to come to common conclusions, but I'm very proud that we can offer today to you this collaboratory. Thank you.
So my name is Jeff Mueller. I'm joining you today from EPFL. I work here in Geneva, but I started the project when we were still based in Lausanne. And I'm going to talk to you about the HBP Collaboratory, which is a scientific hub where collaboration is centered inside in the HBP. So what we expect to happen in the collaboratory is we expect people to be exploring, working, and collaborating, and discovering new science. So this is a place where you can explore the HBP Collaboratory ecosystem, the platform ecosystem, and to work with the integrated web-accessible scientific tools there, and to collaborate with other scientists to gain insights beyond your area of expertise, and to discover the HBP platforms and how they will help you to make your next discovery. Today, anyone can join the collaboratory. So the collaboratory itself is this central scientific hub. It is the gateway to the platforms. The platforms themselves have different access models, but the collaboratory itself, where we try to bring people together, is open to everyone right now. There are some limitations, however. If you are coming in from the HBP, you have the ability to create private collabs or private workspaces that only people that you invite into can share and see. If you are coming in from the community, we only allow the ability to create public workspaces that everyone can view. The collaboratory is a gateway to the platform ecosystem. So this ecosystem has actually been developed by 112 HBP partners in 24 countries in Europe and the world. It's hosted in computing centers throughout Europe. And as I said before, the real value of the collaboratory is actually the access and integration of the HBP platform ecosystem. So the platforms there are providing neuroinformatics, strategic tools in brain simulation, medical informatics, high-performance analytics and computing, neuromorphic computing, and neurorobotics. And each of these has its own, as has been said before, has its own maturity level. Some of them have been developed from scratch over the course of the ramp-up phase, and some of them have been developed over the course of 10 years in a series of European-funded projects. And it's important to note that, in part because of this, but in part also because of the way that we want to engage the community, we have different access models. So Neuroinformatics is public access with one of these collaboratory identity accounts. Medical informatics is the same, so you have access to most of the functionality of these platforms with this HBP community account, HBP identity account, sorry. The brain simulation platform is focused on partnering projects. So we discussed partnering projects a little bit in the morning presentation. So I won't cover those here. And then we have 3 additional platforms where resource constraints are one of the driving factors. And we're following in many cases here from a time-proven model for engaging community with computing resources, which comes from the high performance analytics and computing community. And here we have peer-reviewed access requests. So if you go to the collaboratory and you take a look at getting access, it will describe how to do this in more detail. But here you can see the differences at a high level in the different platforms. And that's it for me. So I'm going to hand off now to Tom Slippert.
Thank you. So my name is Thomas Lippert. I'm leading the High Performance Analytics and Computing platform. So this platform consists of machines, as you can imagine, and the machines that you see here, so-called supercomputers, are belonging to the largest and most powerful ones, on the one hand, in Europe. So this, I can safely say, is probably the most powerful supercomputer, set of supercomputers that can be assessed by a specific community, like the community of neuroscientists who want to simulate and who want to analyze the brain. So both aspects in the name of HPAC are represented. So one aspect is that it is required to have very large memory and very large data capabilities. It's gigantic what you need in order to go to the level, let's say, of micrometer resolution of the brain. So this gigantism is not just la polar, it's just necessary. You can imagine that a full brain finally needs some petabyte, and peta means 10 to the 15 bytes of storage in order to be represented fully. So this is one of the aspects. The other one is the simulation aspect. Machines of this kind belong also to the fastest ones. These are parallel computers. And what you see is machines in Barcelona. So one is really installed in a church, as you can see. That's a very specific, interesting aspect. A machine in Jülich, the first one, supercomputing in Germany. Then comes the CSCS center in Lugano, and there's also the machine at Cineca in Bologna in Italy, and others will come. So most of these sites have more machines. For instance, the machine in Lugano is also complemented by the Blue Brains machine, also a so-called IBM Blue Gene system. So this is one aspect of the machine aspect, the storage aspect, but another one is the software aspect. The software that operates this machine has to be adapted, especially to the needs of the Human Brain Project simulation activities and analytics activities. This comprises workflow systems that allow the researcher to interactively work with the type of problem that has to be resolved. And this is one aspect of the software. The other one aspect is that this has to be supported by high-level support teams. Those teams then make it very effective for the researcher to work on the machine. And as we had heard before, through the collaboratory, the neuroscientists have access to those machines and can use those machines very effectively in a collaborative manner, also with larger research groups working together. Finally, of course, all this has to be represented. I make it simple here. It's altogether, of course, a very difficult process very often, and that's the reason for support, but it has to be represented. And also within the Human Brain Project, we have a large variety of capabilities how to visualize what has been, so to say, simulated, to visualize what has been analyzed, and finally to make it really accessible in an optical manner through our visualization systems that you see on the last row of these pictures. How do we approach now the question or the need that we include the needs of the neuroscientist community in order to form our infrastructure? I think that's a crucial point here. We are not just driven by the machines, we are driven by neuroscience. And so this is finally realized through the so-called co-design projects. These are projects that do not just, let's say, try to solve a problem, but these projects are interwoven. The machine and software development tries to influence, so to say, the way the neuroscience simulation goes forward and vice versa. So that's here as exemplified with the dark blue fields. You see, for instance, 2 simulation projects, and you see also which type of our sub-projects are involved, but there are also the data analytics projects, and others are to come. Yeah? So finally, we want not just to do this from inside. The next step will be that we will look for co-design partners from the outside of the Human Brain Project in order to get all necessary aspects in, and in this way we try to develop our infrastructure. Thank you very much. So. Thank you.
Good afternoon. I'm Sean Hill. I'm a teacher, the professor here at the EPFL, leading the work on the Neuroinformatics platform, co-leading the Neuroinformatics sub-project with Sten Grillner of the Karolinska Institute. So what I wanted to give a sense of today is what the big challenge is in neuroscience, and also what we're trying to address with the first version of the Neuroinformatics platform. And it's not that. So the challenge is that there's a tremendous amount of data being produced all around the world. It's extremely diverse, and it's gathered using many different techniques. And what we ultimately want as neuroscientists to be able to do is put those pieces together, integrate the picture of the brain, so we understand the relationship, for example, of genes all the way through the cells, the circuits, and to the whole brain scale, so that we understand how do all of these levels fit together to produce who we are as human beings. Another part of the challenge is that this data that's being produced is distributed all over the world. It's being produced in large-scale brain initiatives in the United States, in China, partners and collaborations all around the world. And we don't want to have to move all of that data. As we learned before, this can be petabytes of data, and it's simply not viable to move all of that data around. So we have to deal with the fact that the data is not only very heterogeneous, very diverse, but it's also scattered all around the world. So then it's also multi-scale. And the challenge here is, as we try to put together an integrated picture across all of these different scales of the brain, from the subcellular and genes to cells and circuits and so on, as we try to put that together and using brain atlases as an integrative approach to putting this data together, the fact is that our neuroscience knowledge is still rapidly in flux. So it's a huge challenge to actually build a database when the very definition of the concepts that you're using to organize your database are changing. So it's like putting together a puzzle of the brain where the puzzle pieces, their shape keeps changing. And so we had to design a system that could handle that could handle that dynamic nature of knowledge about the brain. And that led us to the Neuroinformatics platform that we're releasing as a prototype today, but rapidly evolving this over the coming months. The data space is the first component. This is really the data federation. So this is a mechanism by which you can access data that's distributed around the world. The knowledge graph is a semantic system. So this uses the latest web technologies for linking the common terminology to the data and registering and keeping track of all the data. Where did it come from and what has happened to it, so that you can search it. It's basically, it forms the heart of the search engine. The brain atlases provide the spatial and coordinate system and the names of the brain regions into which we integrate the data. And the knowledge space provides an essential link between the concept and the concept as it's defined today, and all of the data available from around the world, data, models, and literature, that allow us to see, has that concept, has the definition of that concept changed? How has it evolved? And we keep a living review article about the definition of that concept while maintaining the link to the current data, models, and literature. And all of this is accessible through what are called APIs, application programming interfaces. And that's really essential because we want the community to use these, the platforms to be able to build new applications that take advantage of these capabilities. And this is really what the Neuroinformatics platform is all about.
Yeah, so I'm Jeanette Helgren Kotaleski from KTH in Stockholm, and I will provide a brief overview of the brain simulation platform. So as you know, it's opening up today, and there one can find software and collaborative or strategic modeling workflows, and also models and modeling components. And as you know, one key aim of HBP is to simulate the brain. And we see simulations and modeling as very useful tools when integrating this vast amount of data that is out there and making sense of those data. And we already know that simulation-based science has had a big impact in many fields, including biology and medicine. But of course, to simulate the brain is a little bit of a challenge, since it is the most complex object that we know of. So to simulate the brain, we have to do it in a collaborative manner. And if we are going to do it in a collaborative manner, we really have to have compatible collaborative modeling workflows and strategies and software tools that help us to speed up and automatize this process. And these modeling strategies or modeling workflows, they have to be able to deal with the fact that even though we have this tremendous amount of data out there, it's still sparse data, because these data only represent a tiny fraction of everything that could potentially have been quantified or measured in the brain. So these modeling workflows starts out with sparse data, and then we use principles and all the knowledge and expertise we have to come up with a more dense model that more closely represent the real system. And this work started several years ago in the Blue Brain project, and now it has continued in the Human Brain project in the ramp-up phase. And already the approaches and tools and modeling workflows in the brain simulation platform has helped us to understand better the uniqueness of human neurons, or how to switch between dynamical brain states, to interpret experiments better, and we will continue to try to predict structure and dynamics and eventually functions from this sparse data. But for that, we really need help also from the community, otherwise it doesn't scale. Thanks.
Richard, you're next.
Good afternoon. I'm Richard Frikowiak, and I'm currently attached to the École Normale Supérieure in Paris. I've been responsible for the Medical Informatics project. This project really started because of the fact there is an awful lot of data about medicine in university hospitals and public hospitals which is unused. It's protected for many reasons, including one which interests everyone, which is privacy. So the strategy we took was to bring together professional computer scientists and informaticists and doctors and try to specify something from below up, which is what happened. And so we have developed an infrastructure, which means software essentially, that protects privacy, that allows access to the data on these databases in various hospitals in a distributed manner. By distributed, I mean the sort of thing you do when you ask Google for something. In other words, leaving the data where they are completely eschewing data warehouses and leaving the responsibility for all the privacy and protection with the security officers in the hospitals. No data about individuals leaves the hospitals at all. So this makes access to a large amount of data, which is very dirty, it's incomplete, it has different scales and so on, just the sort of stuff on which data mining works particularly well. And so one of the things that we want to do, and we have started, is to transform the current catalogue of diseases, which depends on symptoms and syndromes and physical signs, into a catalogue that systematically also takes into account all the accessory investigations, most of which are biological, anatomy from scanning, physiology from EEGs, and all the other things. So that's one big piece of work, mainly done by the informaticists once we'd set up the specification, and continued now particularly in the neurodegenerative diseases as a test case, those leading to dementia in particular. The second thing that we've been working on is analytics. In other words, how to investigate these data, how to mine them on the one hand, but on the other hand also how to pose hypotheses and see whether we could do things such as epidemiology, what is the distribution or the incidence or the prevalence of a disease in different countries at different ages, in different places, of importance to public health, so public health research as well, types of research that don't depend on individuals. And that's the strategy of working on aggregated data, which protects privacy, is embedded in the ability to look at these aggregated data in a way that is of particular interest to epidemiologists, public health researchers and such like. And so this sort of work has started at CHUV, which is the Centre Hospitalier Universitaire Vaudois in Lausanne, in the Laboratory of Research in Neuroimaging, which has a strong tradition of interdisciplinary work and sits in the Department of Clinical Neuroscience. And there, the university hospital has been very collaborative, and we have shown that this works, and we have done analyses, and we have installed the infrastructure, and everyone is content. We have now recruited 4 other hospitals to form a group of 5 research and development hospitals for the next 2 years. And they will be specifying the functionality and all the problems that they find in relation to these issues of privacy, security, and all the rest, also the analytics and so on, so that at the end of the second phase, we have the infrastructure which federates the data in 5 major European hospitals and then allows us to integrate it with the analytics to answer significant questions in medicine. Our interactions are primarily with the ethics group, for obvious reasons, with the simulation group and the neuroscience groups, because of what they're doing in terms of associating different parts of the brain with different functions, in association with the simulation of the brain, because in a sense we are also simulating diseased brain, and so we'll be able to put parameters into each other's models to test them one against the other. Thank you very much for your attention.
Morpheus is the god of dreams. He can appear, as you all know very well, in any form of shape during your sleep. Neuromorphic computing is a computing where you copy aspects of the shape and the function of the brain into a new kind of computing architecture. I'm Karl-Heinz Meyer from the University of Heidelberg, and together with my colleague Steve Furber from the University of Manchester in the UK, I'm leading the development of neuromorphic computing, in particular the Neuromorphic Computing platform in the Human Brain Project. Now, if you want to build computers that are like the brain, or at least have aspects of the brain, you have to start from scratch. You cannot really use what is available in your next supermarket, where you usually go and buy a new computer and then write software. If you want new computer architectures, you have to start from the single transistor. That means you have to sit down in your lab, draw the circuit diagrams, think where the electrons flow through your silicon, invent an architecture, build it, integrate it into a system, and develop what you would probably call an operating system, so that even non-experts can use it. You are using your laptop without knowing the intricacies of the microprocessor that actually runs the system. And we have done the same thing in HBP, but of course not only in HBP, because if you want to go from a transistor to a full system, including all the operational software, it takes a long time, certainly more than 30 months. That is the time of the project so far. So the project started basically for us around 2005 with the Spinnaker project in the UK, United Kingdom. That was a national project funded by UK funding agencies, EPRSC. And that is a project run by Steve Ferber from the University of Manchester. Steve is the computer scientist that actually contributed to the design of the microprocessor that you all have in your cell phones, which is called the ARM architecture or the ARM processor. And the idea behind the Spinnaker project is to just use many of those processors and to let them run in parallel through a very efficient network architecture. And that already on its own is brain-like, because it's the parallelism. Rather than having many brain cells, you have many very cheap processors. These ARM processors basically cost nothing. You just have to build many of them, then it costs something. And in particular, if you build a working infrastructure from it, it starts to be kind of expensive. But the processors themselves are very cheap. What you see here is a chip that has been developed by the Manchester group. Actually, the real ARM chip, the real Spinnaker chip is underneath. And that contains 18 cores of the cores that you have in your cell phone. The other approach is the BrainScales project. It has been funded by the European Commission since around 2000 in the Future Emerging Technology Program. This is what we call a physical model. So it's not working with numbers anymore. It's not running programs. There is no code. But it's a model where you basically say, well, rather than moving ions through my cell membrane, which I do in my brain, I just move electrons through transistors. But otherwise, I try to do the same physics. All right, this is why we call it a physical model. And the base chip, which is the basis for our human brain project work, you see on the right side of this slide. Now, what have we done in HBP? We actually scaled it up. We scaled it up to machines which you see here, one in Manchester, now featuring 500,000 of these cores, and one located in Heidelberg in Germany, featuring 20 what we call wafer modules with 1 billion synapses and about 4 million neurons. Now, these machines look a lot like normal computers, like the ones you see in computing centers all over the world, but they are pretty unique. There's basically nothing else in the world like that, with one exception. That's a development done by IBM in the USA, in the IBM Research Lab in Almaden. It's the True North chip. And together with the True North chip, these two architectures are the two only really large-scale neuromorphic computing systems that exist in the world at this very moment. Now, these are large systems. We are making them available to neuroscientists. Why should neuroscientists be interested? Well, one aspect is execution speed. Why is that important? Well, if you think of your own brain, it takes you days or months or years to learn or to develop. To simulate that on a computer is difficult because computers typically run slower. So to simulate a day, you may need 1,000 days, which is 3 years, which is not very practical. The system on the left, the Spinnaker system, runs in real time. That is a day. And the system on the right is an accelerated system. A day is 10 seconds. So we believe that these systems will provide access to the development of the emergence of function of brain circuits, which is a very important research topic. Of course, the other direction is going from neuroscience to computing and to provide an infrastructure for cognitive computing. If you don't want to use these large systems, which are accessible through remote access, through the HBP collaboratory, of course, we have also developed little toys, which we call Neuromorphic at home. These are little boxes that you connect to your laptop. Look at the connectors there. They are USB connectors. And you connect it to your laptop, and you can play with small-scale neuromorphic systems. Why are we doing this? Well, it's a new way of computing, and it's not completely straightforward to use it. Yeah, if you learned how to do computing at school, at university, it doesn't mean you know how to do neuromorphic computing. So it's important that we also take care of training and education. So we think that this, for example, will be interesting for schools and universities, and this is why we did this kind of development. We are looking forward to make these systems available to other users, and this morning Steve Ferber from Manchester said he's not even so sure what to do with these systems. And I think it's good news. Of course, we have some quite good ideas, but I'm sure that a lot of new applications will emerge, and only with the help of external users. Thank you very much.
Hello, my name is Marc-Oliver Gewaltig, and together with Professor Alois Knoll from the Technical University in Munich, I co-direct the Neurobotics platform of the Human Brain Project. We have seen many platforms that deal with creating models of the brain, but how ultimately do we know that these models are actually doing what a brain is supposed to do? In the end, we have to bring the brain models that we built back into their natural habitat, and that would be a body. And the Neurobotics platform allows researchers to do just that. Take brain models, connect them to virtual and real robots, and run them, experiment with them in environments that are simulated or real, to study their behavior and to see whether they actually do what we expect them to do. This is the work from a large number of researchers throughout Europe. It's part of the Human Brain Project. And a major part of the Neurobotics platform is literally to build a virtual laboratory, virtual reality for robots, in which scientists, brain scientists from various disciplines, can test their brain models and can test their models of processing in the brain. A big aspect here is the collaborative part. That is, one researcher can start from an experiment that another researcher has already prepared, if they choose to do so. Or they can invite researchers to join the experiment so that they are together able to investigate what is going on in a particular brain model. Now, a brain model is not a brain model. There are different flavors of it, and we have two extremes. One are the very cognitive AI-like brain models that we find, and this is illustrated here in the top part of the figure, and the other are the brain models which are derived from data. And an unsolved question nowadays is how to bring these two worlds actually together. And the Neurobotics platform aims at making this task much easier, because it allows us to have theoretical models being formulated and let them meet the biological realities of real brains, the real number of neurons that we find, their properties, their arrangements in a real brain. The Neurobotics platform provides a number of tools which guide researchers through a typical workflow that you would have in these experiments. And these tools allow you to formulate an experiment with a brain, a body, and an environment, connect a brain to a body model, a robot, for example, or a virtual mouse, and then do interactive simulations with these components. This is then structured into specific components, which lets you design this. And if you go to the link of the morning session, you can watch a demonstration of the various features of the Neurobotics platform to see where the technical details are and what the possibilities are for the researchers and the research community. Thank you very much.
So good afternoon, everybody. My name is Francesco Pavone, and co-leading together with Marc-Oliver Gewaltig the co-design projects.
number one. What are the co-design projects? These are horizontal actions in the next phase after the ramp-up phase. And in particular, I would like to describe you this co-design project, which is devoted to the development of whole mouse brain model and related mouse brain atlas. So the co-design projects will help to shape, improving, refining, and adapt platforms with user needs. And so the overreaching goal in this case of this particular co-design project is to develop a mouse brain model, whole brain, and will exploit real data, experimental data, which will be produced. And then this co-design project will interact with almost all the platforms in order to use their features, to shape their features, and together with the end user to develop the features in order to realize some data managing, some modeling, and finally some simulation. Then we will use the simulation to validate the data as well. We will concentrate on two use cases. And the two use cases, the first one will be devoted to the study of the motor control and learning. In particular, we want to understand how motor learning shapes activity and functional connectivity. And in order to do that, we will develop a simulation of healthy morphofunctionality into the brain. We will use then this kind of simulation to understand the pathology in the second use case, where we are going to study the rehabilitation after stroke. In this case, we want to understand how functional connectivity is shaped during post-stroke rehabilitation. The real data that we will acquire will be multi-level data, 3D morphofunctional imaging of the mouse brain. In particular, on the functional aspects, we will record some large area calcium imaging which reflects the functionality on the cortex of the mouse, and we will correlate those data with the structural data. One of those will be, for example, the entire reconstruction of the vasculature, as you can see here, of the cell distributions, of the synapses distribution, of the neurotransceptors, and so on the whole brain. But we will take advantage also of some other data, like the whole cell mapping all around the brain, expressing the plasticity and the functionality of single cell with a resolution which is 1 billion times higher than the nuclear magnetic resonance. Finally, we will exploit the very novel field of optogenetics, which help us to dissect the functional connectivity. The end deliverables of the co-design project, which is not a scientific project, the co-design project releases at the end some instruments that every end users can use, developed together with the end users. The first will be a reference setups of the experiments which other people can use. The other three products will be applications, like a virtual anatomy, a virtual imaging, and a virtual behavior application, which will help end users, the first case, to select, for example, in a predictive way, some regions of the brains the users want to work. In the other case, to simulate some imaging experiments or to simulate some behavior. Last but not least, very important, the last application which allows a data explorer and importer and to manage data and to align our data with other metadata and external data coming from other sources like other initiatives all around the world. So other application will be uploaded also by the external users in the second phase. So there is a very strong integration between the platforms, the data generation, the ingestion, and the processing of data. The data can be generated, strategic data, inside of the Human Brain Project, both on the structural and functional aspects. But also, we will take data all around the world, and we will superimpose, by means of atlasing, the structural data, superimposing to the functional maps. Those data will be used for the virtual anatomy, but also for simulation. to create virtual imaging and virtual behavior. Those kind of imaging and virtual behavior will be used also for validation of experimental data and modeling. So, to close these sessions, I would like to give you some resume some take-home messages all around this kind of talks previously expressed. The HBP has completed the first phase of development of the its6 platforms and is now making the platforms available to the scientific community. The HBP platform provides collaborative research tools for brain research and brain-inspired computing technology that will become a European research infrastructure. The HBP platform will stimulate the creation of a global scientific community around the developing research infrastructures. The HBP will continuously improve, refine, and adapt the platforms in close collaboration with the users inside and outside the HBP. The platforms are now available throughout the HBP Collaborative Portal. The HBP platforms have been made possible throughout a collaborative efforts involving more than 800 scientists and 112 institutions and developers in 24 countries. And in the next phase, the co-design project will further develop with users the platforms. Thank you for your attention.